Hello and welcome back. Today we're starting Chapter 8, Inheritance, Genes, and Chromosomes. We will learn about how traits are inherited and some other interesting things about genes. So let's get started. By the end of this chapter and our next lecture, we should have enough information to answer this opening question. Why is hemophilia inherited, and why is it more frequent in males? We won't find out until the next lecture, so please hold your suspense. Starting our chapter about inheritance, we need to talk about the scientist Gregor Mendel. He was a monk in the 1860s that used the scientific method in studies of garden peas. His findings helped support what was called the particulate hypothesis. In his time, they didn't know about genes, but they had ideas that something was being passed between parents and offsprings. They called these determinants, otherwise known as genes today. He experimented with garden peas because of how easy they are to grow and manipulate. Pea plants have a variety of traits that we will see in a minute. He was able to take pollen from pea plants and cross it with another in his experiments. Here's a general picture of a pea plant flower. The stamen is the male sex organ where pollen is released, and the pistil is the female sex organ where pollen is caught to fertilize and make seeds. Pea plants were a good test plant because they had observable physical characters like flower, color, seed shape, and height. Traits are forms of these characters like purple flower, wrinkled seed, and tall for height. Mendel worked with true breeding varieties, meaning that if you cross them with themselves, all offsprings had the same traits. The picture you see here is a Punnett square. You will learn about them soon. When Mendel did his experiments, he denoted the parental generation as P. The resulting offspring from the parents is called the first filial generation, or F1. If the F1 plant self-pollinated, they produced the second filial generation, called F2. You can see in the Punnett square example above that the P generation had one yellow pea plant and one green pea plant. Their offspring in the F1 generation were all yellow, the green trait disappearing. After the F1 generation self-pollinated with themselves, their offspring had the yellow trait, and the green trait reappeared. In the first experiment, Mendel crossed plants differing in just one character, producing what are called monohybrids in the F1 generation. They are called monohybrid because he was only looking at one character that could differ between offspring. The F1 monohybrids were then allowed to self-pollinate to form the F2 generation, what is called a monohybrid cross. You notice that one trait of each pair disappeared in the F1 generation and reappeared in the F2 generation. You also notice that the F1 offspring were not a blend of parental traits, such as being yellow-greenish, but only one trait was present and dominant. You notice then the missing trait from the F1 generation would appear in the F2 generation again. Mendel did this with different varieties, with 7 contrasting traits. He noticed he got the same trend, where one trait would disappear in the F1, only to reappear in the F2 generation. Mendel called the trait that appeared in F1 generation and was more abundant in F2 generation the dominant trait. The other trait was recessive. He noticed the ratio of the dominant to recessive in F2 generation was about 3 to 1. So for every 4 pea plants, 3 would have the dominant trait, while 1 would have the recessive trait. Mendel proposed that these genetic determinants, or again what we know as genes today, occur in pairs, and these determinants, or genes, are segregated in gametes. This meant that each plant had 2 genes, with one from each parent. He came to this conclusion without knowing anything about chromosomes or meiosis, which is pretty remarkable if you think about it for a guy looking at pea plants. We now use the term diploid to describe having 2 copies of a gene, and haploid as having 1 copy. Mendel concluded that each gamete contains 1 copy of each gene, and were haploid. The resulting zygote contains 2 copies, and are diploid. Since he didn't know about genes and chromosomes, he called these alleles. An allele is a different form of a gene that can give rise to a different trait. We now use what is called phenotype and genotype when talking about traits. Phenotype is the physical appearance of an organism. An example would be round seeds or wrinkled seeds. The genotype is the genetic makeup of the trait. It's usually denoted with letters such as big R and little r. Capital letters designate that this is a dominant allele, while lowercase letters designate a recessive allele. The phenotype, round seeds, which is a dominant trait, can be the result of 2 different genotypes, big R, or big R, little r. True breeding individuals have 2 copies of the same allele. They are homozygous for the allele, such as little r, or big R, big R. Heterozygous individuals have 2 different alleles for the same trait, such as big R and little r. Here's a diagram of a cross between 2 parents in the resulting Punnett square. One parent has the phenotype round seeds with the genotype big R, seen here on the top left, while the other parent has phenotype wrinkled seeds with the genotype little r, represented here on the right. Crossing them together results in the F1 generation. To do this, you make a Punnett square. Each parent can only give 1 gamete to their offspring. So you have the round parent with the big R on the top, and the wrinkled parent on the left with little r. One allele goes to each box. During fertilization, those R's will come together to produce the offspring. The F1 generation only has big R and little r pea plants, with the phenotype all being round, because round is dominant to wrinkled. In order to do this, you need to carry the allele from each parent and write it in the box so that each box has 2 alleles. For instance, for the top left box here, the big R from our round parent comes into the box, while this little r from the wrinkled parent comes in the box as well, making it big R, little r. You do that for each box, fill them in. So again, each box gets 1 allele from each parent. For the F2 generation, you cross the F1 parents, which are big R and little r. In here, you can have the genotypes big R, little r, or little r. Majority of the offspring are going to be around, but the wrinkled phenotype will appear again. So that's how you do a Punnett square in a nutshell. Mendel did a ton of these. Here's our first progress check for the chapter. So please take your time to answer these questions and move on with the lecture when ready. After analyzing his results, Mendel came up with two laws. The first law he called the law of segregation. This is when two copies of a gene separate when an individual makes gametes. According to this law, each gamete only receives one copy of the gene. Gametes from an individual with the genotype big D will only contain big D, while a genotype with a little d, the gametes only contain little d. If you cross these two individuals together, as you can see here at the top, their offspring in the F1 generation will be big D, little d. The F2 generation will have offspring with big D and little d. So their gametes will all be big D and little d. The genotypes of their offspring in the F2 generation can be predicted, which considers all possible gamete combinations. When crossing two heterozygous individuals in F2 generation, the Punnett square predicts a ratio of 3:1. Mendel's experimental values were remarkably close to this for all the traits he compared. Genes, as we will learn, are relatively short sequences of DNA found on chromosomes. You can see in the picture above that this is a homologous chromosome pair. It has the trait for a flower's color. One chromosome is from each parent. One of these chromosomes has the allele for a purple flower, while the other is for white flowers. Today we can picture the different alleles of a gene segregating as chromosomes, as we saw last lecture during meiosis 1. Quick review. Does anyone remember what stage of meiosis 1 that occurs in? Here we see the diploid parent cell at the top, the alleles big R and little r. After the DNA is replicated during the S phase of interphase, the cell will eventually undergo meiosis with 4 new haploid cells. 2 of the cells will have the big R alleles, and the other 2 will have the little r alleles. Genes determine phenotypes by producing proteins with particular functions. Dominant genes will be expressed over recessive genes, as we saw in our Punnett squares. Recessive genes are not expressed, but they may produce a non-functional protein. The recessive wrinkled P phenotype is due to a lack of starch branching enzyme, which is essential for starch synthesis. To determine if a trait was homozygous dominant or heterozygous with the recessive allele, we would verify this hypothesis by doing test crosses. We would cross the F1 generation with a known homozygote which had the recessive gene. If the plant in question was homozygous, all offspring would show the dominant trait. But if it was heterozygous, half the offspring will have recessive trait. Here we have an instance where we have a pea plant showing the dominant trait. We don't know if it's heterozygous or homozygous dominant. We know the phenotype, that it's round, we don't necessarily know the genotype. By crossing our pea plant with a known homozygous recessive pea plant, one of 2 scenarios may happen. If all our offspring are round and show the dominant trait, we know that the unknown pea plant was homozygous dominant. If half our pea plants are round and half are wrinkled with the recessive trait, then we know that our pea plant unknown is heterozygous and carries the recessive allele. Next, Mendel tested whether genes were distributed independently in offspring. To do this, he looked at 2 traits and crossed pea plants that differed in 2 characters, such as seed shape and color. He had true reading parents, big R, big Y, big Y, and the other parent with two little r's and two little y's. Big R, big Y, had the phenotype round yellow seeds, while little r, little y had phenotype wrinkled green seeds. In the F1 generation, all the pea plants showed the dominant phenotype, round yellow, the genotype being big R, big Y, little y. He then crossed this F1 generation in what is called a dihybrid cross, since he is looking at two traits. Mendel examined if the traits in the gametes produced by big R, big Y, little y, would it be linked or segregate independently. These two traits were linked, meaning they were on the same chromosome. F2 generation would have 3 more times round yellow than wrinkled green, the ratio being around 3 to 1. If the traits for roundness and color are on separate chromosomes, F2 generation can have 9 different genotypes and 4 phenotypes in 9 to 3 to 3 to 1 ratio. You can do the dihybrid cross yourself, or just take a look at this diagram where it's already done. But his results showed that these traits separated independently, meaning they are not on the same chromosome. His results showed the 9 to 3 to 3 to 1 ratio, meaning that out of the 16 pea plants, 9 would show both dominant traits of being round and yellow, 3 would be round and green, 3 would be wrinkled in yellow, and only 1 would have both recessive traits, being wrinkled green. His dihybrid cross supported his idea alleles of different genes assort independently during gamete formation. This is his second idea, which he called the law of independent assortment. This law does not apply to genes that are near each other or are on the same chromosome, but chromosomes do segregate independently. Here we have a cell that is heterozygous for 2 traits. There are 4 chromosomes, 2 homologous pairs, with 2 traits between them. The alleles are big R, and big Y, little y. Depending how they line up in metaphase 1 of meiosis determines to which daughter cell they go to. At the end of meiosis, you will have 4 daughter cells. This diagram here shows different combinations that can play out. You have a cell with big R, big Y, little r, little y, little r, big Y. Well, you get the idea. You have all possible combinations between the R's and the Y's. Mendel was able to come to these conclusions because of his large sample size. By counting the many pea plants, he was able to see clear patterns. Because of his work, geneticists began using probability calculations to predict expected phenotypes and genotypes. Alright, full disclosure. Next few slides will have a little math, so I apologize in advance. We're going to talk a little bit about probability and how probability is useful when trying to determine the chances of receiving a particular allele. The improbability of an event is certain to happen, it has a probability of 1. The event cannot possibly happen, it has a probability of 0. All other events have a probability between 0 and 1. Two coin tosses are independent events. Each will come up heads half the time. The probability that both coin tosses end up heads is 1/2 times 1/2, which equals 1/4. To do this, you use what is called the multiplication rule. It is used for the probability that two independent outcomes occurring is found by multiplying the individual probabilities. For a coin toss example, you multiply the probability for each coin. In this instance, if you hit heads 2 times in a row. You can also do this with alleles that are dominant and recessive. The probability that two heterozygotes have a homozygous recessive offspring is 1/4. Usually we do Punnett squares to figure this out. But we are really looking at the probability that this individual will get a little r from each parent, which is 1/2 times 1/2, which equals 1/4. After self-pollination of the F1 generation, big B, little b is the probability the F2 offspring will have the genotypes big B, which is 1/4, since they have 1/2 chance of getting a big B from each parent. Again, we usually do Punnett squares to figure this out. But you can do a little bit of math if you want to. To figure out that the probability that offspring will be heterozygous, you need to use the addition rule. There are two different ways with big B, little b, or little b, big B to be heterozygous. The addition rule is the probability of an event that can occur two or more different ways. You add the sum of the individual probabilities of those ways, which is 1/4 plus 1/4 equals 1/2 of a chance being heterozygous. At this point, your eyes may be glazing over with probabilities. If you can do a Punnett square, you can easily figure out probability of receiving a trait. And for most of my questions that will be on either a quiz or exams, you do a Punnett square, you can normally figure it out. So enough about pea plants. Let's talk about inheritance of traits in humans. Human pedigrees can show Mendel's laws. However, unlike pea plants, humans have few offspring, so human pedigrees do not show clear proportions that pea plants show. Pedigrees can show patterns and be used to determine if an allele is dominant or recessive. The above pedigree shows a pattern of inheritance if a rare allele is dominant. Circles represents females, while the squares represent males. Dark color shows that allele is being expressed. This pedigree shows that every person with the abnormal phenotype has an affected parent. Here is another pedigree with a dominant trait. It is being passed generation to generation. And you can see that this trait is generally passed on to the child if the parent has the trait. This pedigree shows a pattern of inheritance if the rare allele is recessive. Affected children often have unaffected parents. Parents are heterozygous carriers, and both parents must have one copy of the recessive allele in order for the child to have it. Okay, here's our last progress check. Last question asks you to do a Punnett square, which you can see on the last slide. Here's the Punnett square problem about dog breeding. Do your best to answer these questions. Let me know if you have any questions about the material in this lecture. Thank you for listening, and have a good rest of your day.